When Dr. Alzheimer himself, over 100 years ago, looked at brain tissue, he said, this is a very peculiar kind of dementia. It doesn't include everyone. This is a particular kind of dementia in which when I look under the microscope, I see these things that are called plaques and tangles.
Well, Joe, thank you so much for taking the time to have this conversation. I've been so looking forward to it.
Hi, Brandon. Joseph Palumbo, and I'm really glad to be here. I'm the Chief Medical Officer and the Head of R&D at BioVie Pharma. We're a small biotech with headquarters in Nevada. We have folks working in the lab in San Diego, and I'm located on the U.S. East Coast.
Yeah, lovely. Joe, maybe give me just a thumbnail sketch of your career. How did you end up at BioVie doing this work?
Well, it's been a wonderful trip. And so I started out as a psychiatrist. I had done my training at Yale and New Haven and medical school in Washington, D.C. at George Washington. And I had always been interested in research, interested in brain function, had worked in laboratories, had done work in nerve system degeneration and regeneration. So I had an absolute fascination with how the brain works and the anatomy of the brain. Also was very interested in cognition, how people think, how people perceive. And when I was in my residency and fellowship, I had the opportunity to pursue some groundbreaking research, which was tremendous at the time. I had the opportunity to move into pharma after career in academia, which had me on faculty at Yale and Cornell and University of Pennsylvania. Started working in orphan diseases, including ALS. Spent time in biotech early in my career at a company called Cephalon, which was successful in treating disorders in sleep. Moved into French pharma and lived in Paris part of the time. Learned a lot about a number of disorders in psychiatry, neuroscience. Eventually ended up at Johnson & Johnson at Janssen and spent about 10 years there, eventually becoming the global head of psychiatry, the acting head of neuroscience. I happened to be half Japanese. An opportunity came up to work for Mitsubishi. As they were expanding into the United States, I started there working in renal disorders, spent time working in neuroscience, of course, was able to get a drug approved in the treatment of amyotrophic lateral sclerosis, ALS, or Lou Gehrig's disease, and then moved to Japan, where I worked broadly across the entire portfolio, both in commercial and spent 20% of my time in a laboratory again. Which was really a lot of fun to get back to those roots. Worked across all of the disciplines, all therapeutic areas in that company, and then moved back to the States after that period of time. Returned to work I had done in the past in neurodevelopmental disorders. We did some great work there. And then I met Kong Do, the CEO of BioVie, who told me about the molecules, about his vision as a philanthropist for how he wanted to move the company forward. And that was about 4 years ago. And now I'm here. So that's the quick background.
Yeah. So having seen the kind of breadth of large pharma, small pharma, biotech, I'm curious what stands out as being meaningfully different, developing drugs at a biotech versus at J&J, for example?
It's obviously all very, very different. A small company has to be extremely nimble. Your decision-making processes are very fast. You're very much the face of the company for good things and for challenges. And you work closely with a team, with a very small team. So it's fast, it's quick-paced, and you're always involved. You can't really take time or a day off. Big pharma is, of course, very different. Big pharma works in some ways like government. There are a lot of friends to make. There are a lot of agreements to get to. You're spending money, and typically it's a lot of money. It makes things slower. It makes things more deliberate. It means bringing in many more partners. So it tends to be expensive, slower, requires a great deal of a different kind of dedication, a dedication to knowing what's happening in the field and bringing an entire company along with you. Uh, Biotech is it requires many, many, many of the same skills, but you have to be actually able to outmaneuver uh, the larger forces. It's more like working with drones, right? You had to be really small, you had to be accurate, as opposed to running a battleship or an aircraft carrier, because it's those kinds of odds. Uh, Big companies tend to um, move, like an aircraft carrier. They are certain, uh, but they're a little bit slower, but they're enormously powerful. And smaller companies were always looking for a way to be successful, a way to get patients the best drugs, the best treatments as quickly as possible, and do very directly benefit the shareholders. As clearly, these are both businesses, but with a smaller company, you're very much more aware of who's supporting you, and we take that quite seriously.
Do you miss running larger programs and portfolios like you were at J&J versus focusing on individual like assets?
I miss the big teams.
Hmm.
Um, I miss the day-to-day camaraderie of having a group of people who work together well. Um, in those larger organizations, I had set up J&J in many ways as if it were an academic department. We brought in people who, of course, were research oriented, and people who also could more than hold their own uh, with the commercial needs of an organization. It's very easy to be smart. It's much harder to be collaborative. And once you find those exceptional collaborators, and you see them in person and you have lunch with them every day, that's an absolute treat. That's an absolute honor. And you can come up with new ideas very, very quickly when you're in a team like that. Smaller pharma is hopefully you've made a lot of friends, and you hope that those friends will still collaborate with you. So it's a lot more active. The folks aren't just sitting there at your elbow at the lunch table. You have to go out and reach them. So I miss that easy access to lots of people. On the other hand, I've been lucky enough to make good friends over a career, and I reach out to those wonderful folks as ever whenever I can to help within a smaller company.
Yeah. Maybe take me into the inside the story around your work in ALS.
Sure.
I'd love to hear more about what that was like and the kind of process of working in that space.
So when I first came to ALS, I knew very, very, very little about the field. I was a well-trained neuroscientist. I was well-trained in cognition and disorders of the brain, disorders of the mind. But despite the work I'd done in the laboratory, I didn't know motor systems nearly as well, and I certainly didn't know inflammation at the time as well as I did. I've learned over the years. So it took friends, right? It took colleagues, it took mentors. So my first attempt at ALS was working with another wonderful biotech. And again, that company was Cephalon, and they were located in Pennsylvania. And that's one of the, it was a hotbed, the Pennsylvania area of neuroscience at the time. Um, and there were terrific people, including the CEO, a guy who sadly has passed on, but was absolutely brilliant and committed to science and patients. His name was Frank Baldino. And Frank asked me to start working in ALS. We had a program there with at least one positive study. The drug was called myotrophin. And Frank enabled me to go and work with some of the great professors. And there are many people, and I know that I'll leave out some names. One of the people was Dr. Stan Appel. And Dr. Appel had moved from Boston to lead a program in Texas. And he invited me in to spend time with him and learn what he did, uh, to work with patients. And um, he enabled me to learn a lot more than I had. Um, and he introduced me to a group of global experts. Now, ultimately, that program didn't succeed in registration. But when I came to Mitsubishi, I had had that experience um, working in ALS, and it was literally 20 years later. I discovered that there was a program that was ongoing with medical experts in Japan that appeared to have promise. It really wasn't on anybody's radar. So I watched it very carefully. And as the data began to come out, I gathered it uh, with my Japanese colleagues. And I went back to the same group of experts I had met 20 years earlier, because I wanted to make sure that this was a molecule, this was a compound, this was a treatment that would really work. So
Mm.
I asked these folks, Dr. Appel and others, Terry Hyman Patterson in Philadelphia, uh, a group of folks who really, really cared about patients. Does this data look real? Can you say that this is a molecule, it is working? And they said, yeah, it appears to be working. So at Mitsubishi, we put together a team of Americans and Japanese, worked very closely with the US FDA, uh, gathered data, some of which at that time were still on floppy disks and written in workbooks, translated that into English. And then we were successful in very rapidly bringing forward an intravenous product. And shortly thereafter, we knew that the intravenous product was going to be helpful, but we wanted something that was easier to take. So we were very, very rapidly on an oral formulation. We were able to get that approved. But working with people across the country and across the world on something really this meaningful, and there hadn't been a drug to come forward in over 20 years at the time. There was only one, a drug called Riluzole, which certainly was effective in slowing the disease. What we were able to do through the work that we did was we're able to preserve function for longer. And that's hard. That's hard because when you take a drug and you don't necessarily notice that you're getting better, because you're probably not. You don't notice much, but we can look at the course of an illness and know that if you had not been taking the medication, you would have been progressing more rapidly. Um, and people hadn't done that kind of projection often in neuroscience. It would take another 10 years or so before the Alzheimer's community was able to bring forward molecules that slowed progression of disease and preserved function. But we were there early, and frankly, we had a very, very cooperative, cooperative is not the word, a very engaged group of scientists at the Food and Drug Administration in the United States and Japan and other countries uh, who recognized that there was need. US and Canada were particularly engaged in this way, Japan as well.
One of the things you said kind of stuck out to me, which is which was the I guess scientific diligence process you went through, just trying to understand like how real is this data.
Yeah.
I I feel like everyone is doing that, right? Like in biotech land, everyone is trying to do that. What do you think you've seen that points towards, hey, this is truly believable in the earliest stages versus I don't really know if this passes my my kind of sniff test.
Yeah, it's really a very difficult question. And the reason it's difficult is because so many things that look really, really good in the computer or
If it.
really, really good in animals, sometimes doesn't work out in the human condition. And sometimes we never learn why. Um, some of the work I'm doing now, I can probably talk about later, is based on that duality between animals and humans. But to make sure that something's really working, you want to create an experiment set up to answer a particular question. And then you need to be able to replicate that. Right? So FDA typically looks for at least 2 studies, right, to prove that something works. And then they, do this to a degree of statistical significance. In other words, it's only 5% unlikely that this could have happened by chance. So it's 95% certain. Then you do that twice. And then typically they may ask you for additional studies after the drug is on the market. And that's just very much the minimum. What the scientists had done in Japan is they had taken this drug, edaravone, and they had shown that it worked very well in ischemic stroke. And it had become the standard of care in Asia for the treatment of stroke. And there were a group of very dedicated scientists, physicians, and other folks who said, can we try this drug that we know to be effective in stroke and we know to be safe in stroke and see if it works in ALS? And so they did that. And they conducted a trial. It didn't quite work. And then they conducted another trial. They changed the inclusion criteria and the dosing. And they finally got it right. And then they said, okay, let's repeat that experiment. Let's enrich that. Let's confirm that what we saw in this last experiment, prospectively studied, is really true. And they did that. And they did it with a different kind of trial design, which was what was very much enriched for patients who were progressing. When you do things like that, when you do this enrichment, it's important to understand whether or not that enrichment is generalizable to a larger population. Why that's so important in a disorder like ALS is because it's an orphan condition. So you have people who have the disorder. They really want to participate in research. Frankly, they're trying to help others who has a disorder. The ALS community is extremely strong that way. And you have to move pretty quickly. In the past, and this is actually still true in Europe, they wanted studies, the scientific community, they, meaning the scientific community, wanted studies that ran several years, which meant large studies, lots of patients, and it might take 4, 5, 6 years for a drug to be studied. And by that time, the folks who had largely started in the study would have passed away. So you were looking at time, frankly, time to death. What we wanted to do is look at function. And we wanted to try to get these studies to be much shorter. We wanted them to be smaller. So we did things differently. I guess if there's anything that I'm particularly proud of is working with those Japanese scientists to bring this concept of enrichment to the United States, doing faster, smaller studies, and then repeating them in order to get a drug approved. Now, because that was something that was new, I needed to make sure that when I took this to the experts in the community, the people who really knew this disorder, the folks I had met 20 years earlier, did they see what I saw? All right. And that was that extra step. And this meant that the FDA was going to be judging this program based on history in Asia, in stroke, as well as what these scientists had done in Japan, and to enable us to bridge that into the United States. And we got a lot of assistance in figuring out exactly how to do that. We published on these aspects of the disorder and the aspects of the medication. So I think everyone in biotech is trying to do these things. But I will say that for the community and the scientists who are working for me, they were extremely careful because we didn't want to produce false hope.
Huh.
Wanted to be certain that what we saw scientifically and clinically was relevant.
How do you balance the desire to do that with, um, I mean, everybody talks about timelines, right?
Mhm.
And speed and all this kind of stuff. How do you balance the desire to um, really take your time and make sure you've kind of validated everything to your best ability versus maybe the kind of uh, like shareholder like or investor dynamic of wanting you to go as quickly as possible?
Yeah, I mean, some people will say go as fast as you can and no faster. That really doesn't answer the question, right? But people say that, and typically scientists will say that. And I think you really have to have a partnership with the community of experts. They need to understand what you're doing because they're going to help you communicate to your investors. Well, is what these folks are doing, does this just make sense? And then you try to be as open as you possibly can be about your progression. Um, and what you try to do is to develop a molecule to its next logical step. So you're not trying to promise everything all at once. You're trying to say, for this much money, we can deliver this endpoint, right? Uh, and you try to publish on that and you try to do it openly. But it's really development to your next decision point. And the next decision point is typically, have you learned something? Right. And what have you learned that enables you to move on or to adapt? But you're really asking for funding to that next decision point. Um, and I think that's how you do it best. Um, and we've tried to do that at BioVie. We've tried to show, you know, all of our data coming forward, um, and to share what we learn.
I'd love to maybe take a maybe a step back and just talk about the broad landscape of neuroscience right now. Maybe give me an overview as to like how you see the kind of landscape and the current momentum and where you're most excited.
It's never been a more exciting time to be a neuroscientist. Um, and that probably is true every day as we learn more and more about how the brain works, how the body works. I guess the biggest difference is that we're no longer working in the silos in which we once did. That I think there's been a generation of scientists, physicians, researchers who have worked across disciplines, across fields, and I've been lucky enough to be able to do that. Who take what's learned in oncology and say, hey, there's something similar in neuroscience, or what's been learned in immunology. Say, there's a system that looks just like that in neuroscience. And to begin to look at these as systems. And I think when I first started doing this, there was a sort of this concept, how simple of how simple can you make it? Now, I know why we want to make things simple because it makes it testable. But I think there's a growing understanding that the systems biology of neuroscience is incredibly important to everything from neurodegeneration to depression. And as that moves forward, as we see how drugs affect things like DNA methylation, right, which is essentially changing and modulating, how your DNA functions in real time. With this kind of knowledge, things are extremely interesting. So, we have what are called small molecules, which are sort of the typical things that you would think of when you take a pill, like an antibiotic is typically a small molecule. And then there are these large molecules, which target immune systems typically. There are DNA therapies, there are RNA therapies. We have a host of different mechanisms now that we didn't have. And we have different folks looking at all of this very, very aggressively. And I think that makes neuroscience particularly interesting. I guess what I'm most interested in is this interaction between inflammation, between diet, between how we um, exist in our environment, uh, the things we eat, um, and how our bodies work. And in neuroscience, it means we have a greater and greater understanding that there are things that are good for us. Um, and we need to understand why those things are good for us. So exercise and diet matter to the development, and we all understand this is, say, diabetes, but it hasn't been so long ago. We first started to understand, well, this link with diabetes is related to a number of things, metabolic syndrome, but also inflammation. And inflammation and its management is extremely interesting in terms of what the future will bring to us. So, 30, 40, 50 years ago, we began to have this hunch that statins, were important in treating high cholesterol. I think what we understand now is we do a lot of things to our bodies that affect the vasculature, that when that vasculature is inflamed, it's more likely to lead to poor outcomes. It's more likely to lead, for instance, to the development of cholesterol-related illnesses. So it may not necessarily be cholesterol, it may be inflammation. And if we can manage inflammation, high blood pressure, all these sorts of what we think of as disorders of internal medicine rather than neuroscience, turns out that internal medicine is incredibly important to keeping your brain healthy. So when people ask me in general, what are the things I can do for my brain? I can say, well, the things you do for your heart are also really good for your brain. So this, again, this understanding that there are systems that we treat rather than sort of these, I'm going to say, monotonic interventions. And that medicine is medicine regardless if it's the brain or the heart or the body or the gut, and understanding that these things all, they talk to each other and play with each other. And if we can find out how to facilitate those things causing, relating to better health, that's what's exciting about neuroscience right now. And people are moving there in that direction. And I think that's incredibly exciting.
So if I was a young researcher, where would I go? Like where would you counsel me to go to kind of participate in this?
Well, let's say you want to be a physician, right? As a researcher. And I think that's, a path I'll walk down right now. I think the best thing that a physician can do in learning how to be a researcher is to sit with patients and families, to learn the signs and symptoms of disease and not skip on that part. We have a lot of folks who want to just run to the laboratory, and I think that's wonderful. But I think you miss something if you're not at bedside for a number of years.
Good.
It teaches you compassion. It teaches you a lot about the human condition and about how illnesses affect us and how to detect them. So that's sort of the first step is be a good physician. And by being a good physician, it means that you embody all of that. It becomes a calling for you. From that place, I think it enriches your abilities to do research. Now I'm biased. That's the path I took. But I think it's a good path. It helps to understand how medical systems work. It helps to understand how care is provided. So I ran a hospital group, and I actually ran a hospital. So it was important to understand, what are the needs that clinicians and administrators are going to have relative to the patients and families we need to treat and the medications and interventions we bring to bear, because it all, again, is part of a different kind of system of healthcare, and how to make these, how these treatments are both meaningful and credible treatments. It means that today we're developing drugs that we need to show have clinically relevant outcomes rather than, just hitting a p-value, as I mentioned before, statistically significant change. We need to show that these drugs show uh, meaningful improvement or meaningful change in the people who take them to create a certain value, because we do pay money for things. So value becomes part of this. Um, so maybe a longer answer to the question, but I think it. So going into research, become a clinician first, if that's the path you want as a medical doctor. Um, Learn systems biology.
Hmm.
Um, I think it's enormously helpful. People who work as engineers and then who go into medicine do exceptionally well because they ask how things work before
Uh,
they why things work. And there's a subtle difference. So how things work means what are the interactions, and then the why can follow. The why helps to understand how the system works. A typical question I would get from the folks that I help to train when I go to conferences and I'm asked to help mentor people. People will say, I want to be a brain scientist. I say, that's great. What do you want to study? Well, I want to learn how to use an MRI, or I want to learn how to do this kind of imaging, or I'm interested in the brain gut interaction. I said, that's great. What's the question you want to ask? Do you have a burning question? I mean, it doesn't have to be the quote unquote right one, but there has to be a question, because otherwise, I think you really just become a technologist. And there's nothing wrong with technology, but I think you need to ask yourself, what are the questions I'm trying to answer? What am I trying to learn? How am I going to change the practice of medicine? And you'll go down a lot of wrong avenues. That happens. But that is what I hear from. I want to learn how to use this technology, and unfortunately, for that kind of thinking, technology always changes, right? And you want technology to advance and train, but you want to be able to use the technology to answer the questions you have, rather
so,
than
Joe,
sort of,
what are the questions you're trying to answer right now?
the questions I'm trying to answer right now are related to inflammation. It's related to this question of, if you can treat inflammation, how does systems biology change, and how does it make you better? Uh, this question came to me first probably 2 decades ago. I was working at Johnson and Johnson. We were working with these exceptionally interesting and wonderful folks at Emory University, with Charlie Nemiroff and other folks who were there. And the question was, if you blocked TNF, which is this body makes TNF to create and fight infection, right? But it creates a lot of inflammation. So there's a tumor necrosis factor, but it doesn't exist only in tumors. It exists broadly and is a chief driver of inflammation. So we used a molecule called infliximab, but brand name was Remicade. And we looked at patients who had high levels of inflammation. And we saw that when you looked at patients with refractory major depression and you gave them a drug that stopped inflammation, you know what happened? They got better. Their depression went away. And this was this huge learning for the community of, wow. Right? And then I've worked with other drugs that are being developed by other companies, looking at different aspects in this pathway, this cascade of depression. So we're looking at schizophrenia. We're looking at other conditions. We certainly know that if you block inflammation, you can treat certain movement disorders, not just ALS, for instance, we showed when I was at Mitsubishi, but we learned that if you change inflammatory responses, even with natural products, you can treat relapsing multiple sclerosis. That's what Mitsubishi had done in cooperation with Novartis. So, yeah, these partnerships are incredibly important. But the point being, what I'm trying to understand right now is, if you modulate inflammation, can you protect the brain? Right? And what are the pathways by which you do that? How do you measure that? So we're very interested in measuring changes in DNA called methylation. Right? Methylation, again, changes how a pathway will function. So that's what I'm really interested in. So we're looking right now at BioVie at long COVID, seeing can we change the inflammation so that people's brain fog can go away and so they can be less fatigued and function again. So that's a big question we're asking with assistance from the U.S. Department of Defense. They are extremely interested in this. We're asking that question in dementia and Alzheimer's. We're very actively asking that question in Parkinson's disease. We've shown some data in a small study that suggests that when we block inflammation with our drug, we can improve what's called motor function, the ability to move, even the ability to sleep. So now we're asking, can we get in really, really early? And can we provide an assistance and maybe even a first-line therapy? So again, the questions I'm asking are about inflammation, its effect on aging. And we have some early and very interesting information that we've recently published and we'll be talking about in upcoming months on whether or not these changes we're seeing in methylation can influence the course of aging. So really, really important things. As you can tell, I'm really excited about them. We want them to be safe, right? We want them to be, we want these to be treatments that treat inflammation, but don't make us susceptible to infectious disease. For instance, you might think of a steroid and steroids we take, and yeah, they make us feel better, but they can block our ability to fight a disease. So we want drugs that are likely to change our response to inflammation, but not to suppress our immune systems. And again, we're beginning to see that with some of the drugs we're developing.
It's really interesting to hear that, like, that your primary focus is inflammation as it relates to, let's call it, like, neuroscience and psychiatry. Um, is there a particular reason why that's the kind of downstream effect you're most interested in, as opposed to maybe like other parts of the body? Is there like a hypothesis that kind of links these two specifically, or is this just like by nature of your background and training?
It's a lot of all of that. So yes, that's the background and training I have. Certainly, I played baseball as a kid and as a young adult. So I tend to see things in terms of baseball rules. My kids tease me that, I speak of things being home runs and grand slams. So yeah, there's a filter through which you see things as a neuroscientist, as a psychiatrist, as a bedside clinician, as a hospital administrator. It changes the lens through which you see things. On the other hand, having worked across multiple therapeutic areas, in my career, it helps me to look for these links. Um, there are practical matters as well. I mean, if I wanted to take this question about inflammation into metabolic disorders, diabetes, etc., typically these are extremely large studies that may involve thousands of people over perhaps even a decade. And if I were at a really big pharma, I might consider that. But in a biotech where you typically have enough money to get through a year at a time, you need to do things that are just a little bit more focused. And that's in part why we're in these conditions in which the scales in the diseases are more approachable for biotech. And then you hope that what you prove there then gets noticed by other scientists and they have the resources to expand. So there's a mix of things. Certainly, these mechanisms could work in other conditions as well, because inflammation isn't solely in the brain. Inflammation is everywhere, right? Aches and pains are often inflammation, but you have to pick a focus because you only have so many resources. And then you think about, where do you get your biggest bang? And for me, that's in these conditions of neuroscience where there's tremendous unmet need, um, and that need isn't going away.
So maybe let's use that as a jumping off point. And I know we're almost short on time here now. But tell me about the kind of inflammation hypothesis in Parkinson's and Alzheimer's, where you're doing work today.
Let me start by looking at what we've believed that we've known for decades and decades about these conditions and how that's changed. So in Alzheimer's, when Dr. Alzheimer himself, over 100 years ago, looked at brain tissue, he said, this is a very peculiar kind of dementia. It doesn't include everyone. This is a particular kind of dementia in which when I look under the microscope, I see these things that are called plaques and tangles. And he was looking at these in a light microscope. And what he discovered was, okay, so this is sort of the beginning of the amyloid hypothesis. What is causing this? This is tau, this is amyloid. And then that almost went away because we began to think, well, what are the other things that cause dementia? Are these mini strokes? Is this hardening of the arteries? These are all the terms we used to hear about. And we thought of dementia as this very broad thing that we sometimes couldn't even diagnose until after someone had passed. We'd have to look at the brain to discover what kind of Alzheimer's it was. And then about 20 years ago, we began to think, well, amyloid seems to be there a lot. Why is amyloid there? Let's see what happens if we remove the amyloid, see if we can slow the disease. And we've done that. And we've gotten about as far as I think we're going to get with that right now. We could slow the disease a little bit. But we didn't ask what causes the amyloid, right? So typically when amyloid builds, it builds in reaction to inflammation. And amyloid is in part protective against further inflammation. So in a way, it's like a scar tissue, right? So that's what we knew. But we weren't really thinking about what were the processes going on in the brain that led to this really interesting pattern of, and this is another little weird thing, right? Is that Alzheimer's in its traditional state starts in parts of the brain and then grows out. So why would that kind of happen? Well, we had to ask ourselves, okay, what was happening? Why was that happening? And there were tremendous scientists beginning to work on this and saying, there's a process by which the brain becomes inflamed, by which there's a substance called tau, and tau breaks and fractionates, and that starts acting almost like a virus. And we weren't really necessarily thinking of that. So now we're thinking of it. Same with Parkinson's. So Parkinson's is this fascinating condition that we think of as, or we have thought of as simply being this loss of dopamine in the brain. And certainly that is true. We lose dopamine neurons, we lose them in a part of the brain called the substantia nigra, which is basically the black stuff. We're going way back in history. What was not working? Well, the black stuff wasn't there. And those were the neurons that were making dopamine and resulted in moving. But there's something else very, very fascinating. But if you walk up to someone with early Parkinson's and you kind of get them excited about something, they can actually move. And why is that? Well, part of it is there's this, there's a surge in dopamine, but there are also auxiliary pathways that we really haven't thought about, or we don't really pay attention to. And those auxiliary pathways, in addition to the substantia nigra, are all susceptible to inflammation. Right? And now we begin to think in the last 10 or 15 years, is there something that goes on in our guts where some kind of disease, some kind of process comes from the gut, goes to the brain, and causes inflammation, and then leads to dysfunction? And you can think about long COVID. You can think about COVID when people started to lose their sense of taste, lose their sense of smell, why would a virus that infects our lungs affect our ability to smell and taste? And it has to do with inflammation and inflammatory response and the activity of those viruses within the nervous system. And sometimes we don't think about that. I think Parkinson's is similar. I think that Parkinson's is in for many people, starts as an inflammatory disorder. And that the natural molecules in the brain that can, again, work in the systems biology, we can find ways to make them work. So very, very quickly, so how the drug I'm working on, I hope is working, is through this aspect of systems biology. There are similar molecules that exist in the brain that can be modulated to treat postpartum depression, for instance. This molecule we're working with actually is, and this is the really cool thing, it is a naturally occurring brain molecule that our scientists just made a little different so that it's more stable, lasts longer, you can take orally. And so that's my question. Can we get in early, use the systems biology approach, using, frankly, a naturally occurring brain molecule to change the course of these two disorders? Um, and, I spent the last 4.5, almost 5 years working on this experiment. And to this point, I'm pretty encouraged.
Remarkable.
Thanks.
Well, Joe, maybe kind of in closing here, I'd love to hear from you. What do most people get wrong about doing research in Parkinson's and Alzheimer's?
Very few people get things. Okay. If you come to Parkinson's, you come to Alzheimer's, you come to neuroscience, you're here for a reason. And the reason you're here is because you want to get people better. At least I hope that's the reason, right? You want to reduce suffering. That's the most important. If you want to think of sort of an Asian concept, a Buddhist concept, life is really, really, really hard. People suffer. And as scientists, as clinicians, as physicians, as researchers, we want to reduce suffering. So I don't think people really get that wrong. And if they are not coming from that position, that is wrong. I would ask people to think about working with colleagues who are diverse in their thinking, put together really, really good teams, listen to people, listen to their concepts, listen to their ideas, if they sound a little bit wacky. Because I think, this systems biology is helpful in a very serious way. Look to evolutionary pathways. We sometimes don't do that. And I am very, very hopeful as this next generation of researchers goes into the laboratory, into the clinic, that they'll be talking across scientific boundaries. And I think that's the right thing to do. I don't think there's anything wrong. I think there's just a lot more right that we can do.
Yeah, I love that. Well, Joe, thanks again for taking the time today. Really appreciated you joining to kind of educate me here.
Well, Brandon, a delight, and thank you for inviting me.
Of course.